All right, dear guests, it's 5:05, so I think it's a very good time to actually start tonight's event. It's called First, the inaugural lectures of ITU, and actually there's a bunch of firsts, so it's the first event of this type this year. It's also the first inaugural lecture that I'm actually have the honor to host and to welcome you. And it's also the last in the semester, which is, as you know, is approaching its end. So I'm very happy that we brought together a very interesting combination of like research, you as visitors, I see some industry faces, I see a lot of students, so that we'll have a good basis for a profound look into how research at the ITU is actually done, and then afterwards have a promising conversation with rich inputs and then also rich responses from our speakers. I'm very happy that we were able to present today two very promising speakers from an interdisciplinary field of research that I, as a layman, would call like computational medicine, but we will see how this in particular will evolve, and so that we can address your craving for information, craving for input, but also your interest in seeing what actually is done in the part of the research and get to know the actual two highlights of today. I will hand over to our president, Stephanie Lindstedt, who will do a brief introduction before the two talks. Then we'll have, we'll listen to our researchers, and then afterwards we will have the discussion to really engage with the researchers, with you, and talk about what's actually on the plate, what's on the topic, and what we can learn from each other and where we are heading all together at ITU. So with this, I'd like to hand over to Stephanie.
Hello, everybody. I'm very happy to see so many researchers here and whole groups brought here. Perfect. This is the spirit. Well, we are extremely happy that we have two new professors, both in computational medicine, here at ITU. So this is a big step forward in that direction. So far we had two tenure-track professors, Nina Humbig and Ji Mei. So these are now the two senior professors coming in, and yeah, I'm already absolutely excited about the talks to see what they will tell us. Okay, let's start with Spiros, and can you switch to his slide? Yeah. First slide, whatever. Yes. We always do this in the beginning to talk a little bit about the career path of our new professors, so that you have an understanding what's their background, where they come from. So Spiros comes from computer science, University of Bradford, where he did his master's, then went early on into bioinformatics for his PhD, Manchester University, and then he already went to UCL, where he was until we lured him to Linz. So he had a lot of different positions at UCL, senior researcher, senior lecturer. This is the typical UK career path, so that's very different than how things are here in continental Europe. Then a full professor and also the lead of the data science center, specifically, of course, everything with health data, medical data, which he has been working on. He's also an associate or honorary professor at the University of Athens, and so this is kind of back to your roots, I assume, and visiting sometimes there. And now, yeah, since when? October, you're already here at ITU, and very busy already with setting up with the personalized technical medicine initiative, which is going on. And yeah, well, the floor is yours, Spiros.
Thank you. Thank you.
Okay, thank you very much for the invitation to talk and for the warm welcome now and since I joined. So yeah, I grew up in Athens, and I'm now based in the two places that have a lot of rain, Linz and London. And my background is computer science, and I think that's kind of like what I enjoy working about in health, is they can bring kind of this computer science approaches into a domain that traditionally has heavily relied on sort of statistics rather than data science. And I'm interested in all sorts of things that I'll talk about in my presentation. I won't tell you how old I am, but I'll show you a picture of my first computer when I was a kid that had 4 kilobytes of RAM. So today I'll talk about precision and imprecision medicine, about electronic health records, phenotyping, which is kind of my primary research domain, and then some research exemplars that I've picked out of things that we've done with electronic health records in the UK, but also kind of abroad. So the current paradigm of care and research is a one-size-fits-all paradigm. So we have patients that present with a particular disease that clinical medicine treats as homogeneous, and we give the same treatment to all patients. And the same treatment may work in some patients, may not work in other patients, and in another subset of patients, it might develop adverse events and need to switch treatments. And where we want to be, ideally, is what's called precision medicine or personalized medicine, is to understand the heterogeneity between patients and between diseases and the different subtypes, and target individuals with the right treatment and the right intervention at the right time, as to maximize the number of patients that develop a positive effect. And it could be the case that we don't actually have a treatment and we need to identify new targets and develop new treatments for these individuals. So this is a huge problem in clinical medicine, and it's only exacerbated from the fact that people live longer and present with more complex clinical manifestations and multimorbidity that I'll talk about later. How does this look in reality, this sort of utopia, this ideal scenario? This is sort of from a really, really cool review on colorectal cancer. And it shows kind of like the sort of a time lapse of how treatment and therapy works. So from the sort of when this one gene was identified that was then targeted by one drug, to sort of scientists developing a new understanding of genomic subtypes or subtypes of disease that are driven by transcriptomics or immunological subtypes, and in the process developing new biomarkers to measure activity in those subtypes and new drugs. So they went from like a one gene, one drug paradigm to like a multi-gene, multi-drug, sort of multi-biomarker reality. So this is where we want to be ideally for all diseases, not just one disease. Why? How big is this problem? So the problem at the moment is huge. So this is quite an old slide, so the numbers are probably even larger now. But the reality is that it takes billions of dollars to bring a new drug into into kind of circulation. Most of the drug development that happens fails, so over 97% of drug development fails, and usually it fails at a stage where you've already spent sort of $1 billion to do that. And the reality is that most drugs that are prescribed, at least in the US, and these are the top 10 most grossing drugs, so the drugs with most sales, they fail to work in 50 to 70% of the patients. And this is because this one-size-fits-all type paradigm. So why is this happening? I would argue that it's happening because we probably underestimate the heterogeneity of disease, and also that we use oversimplistic representation of diseases in our research pipelines, and as a result in our R&D pipelines. So it's safe to say that most of the decisions in current clinical medicine are sort of derived from a time where data were small and limited in size and resolution. So in cardiology, which is sort of the stuff that I've worked quite heavily in, the landmark study in cardiology is called the Framingham Heart Study, and the findings from the study dictated how clinical care was delivered in the UK, for example, even though it's a study of sort of a prospective study of roughly 5,000 men and women in a small kind of town in Massachusetts. And this is one of the first studies, if not the first study, that showed that smoking increases your risk of ischemic heart disease and cardiovascular disease quite substantially. And they were able to show that actually, this is kind of an established fact since then, and then that kind of gets propagated. And this is what's called a prospective cohort study. So you sort of recruit individuals, you send out letters, you bring them to a clinic, you take measurements, you might send them questionnaires, and things like that. But the reality is it's really, really hard to scale. So they did it in, what was it, like 4,500 men, and women. At the moment, the largest recruited cohort study is probably, that's consented, is probably all of us in the US, which is roughly 1.5 million. In the UK, we have the UK Biobank, that's 500,000 individuals. And they cost anything up to, in euros, I would say, let's say like 100 million euros maybe to sort of bootstrap and run and recruit individuals. So it's really, really hard to scale them. They're very high cost and research intensive. There's selection bias because the people that you'll invite and will reply to your questionnaires and will send data are biased. So you know by definition they don't have severe diseases, or they might have underrepresentation of some diseases. So the big question is, can we use data that were collected as part of healthcare for research? So can we use the data that we all sort of generate when we interact with the healthcare system, but use it for research? Of course, by taking into consideration all the biases that it has. So the fundamental shift, at least in the UK, happened, I guess, like 20 years ago or so, where humans were started being considered as a new model organism, and hospitals are basically large-scale phenotyping centers. So in the tiered healthcare system that the UK has, a lot of data gets recorded that we can use for research, and that data started getting collected. Obviously, when I moved to the UK in 2000, my medical records were on paper, and every time you moved doctor, like they would send like a brown envelope from one doctor to the other, and they would sort of use that to keep your record. So there's been huge sort of progresses into digitized records. That's, again, it's not uniform, but it's happening. So when we interact with the healthcare system, we generate a lot of data and metadata. So in the UK, we have a tiered healthcare system. So everybody's registered with what's called a primary care or family doctor. And compared to Greece, for example, it's different. So you can only go to a hospital if the family doctor refers you to, and obviously unless it's like an accident, an emergency, or things like that. There's no way, like in Greece, to say, today I'd like to see an ophthalmologist, and you just walk to the hospital and get like so everybody's registered with a primary care doctor, and you get regular checkups, and you get a lot of data that get generated as part of those checkups. So these data, these individual data points, they get recorded in your health record that's digital now in both primary care and hospital, and follows you throughout your health journey within the country. That's not to say, unfortunately, that all GPs and all hospitals share the same health record. They don't. But at least when you live in one area for a long period of time, your data is in that area, and the healthcare sort of system can use it. The data gets recorded in what's called the medical ontology. So you may be familiar with things like ICD-10, which is the most common one in sort of worldwide, and SNOMED-CT, which is sort of the de facto standard now, at least in the UK. So imagine these are like really, really comprehensive dictionaries of terms that doctors can choose to record information. And all of these are like the building blocks of the information that gets recorded when you interact with the healthcare system. The problem is that the data that get generated as part of clinical care are not research quality data. So imagine that the Framingham study that I mentioned, they had a really strict protocol. Everybody joined the assessment center. They got their blood pressure measurement at a very specific location in the arm, sitting down. They followed a very, very strict routine that was driven by a protocol that they had quality controlled. Whereas in healthcare, data is very, very fragmented. Every doctor does things slightly differently. And it's very, very important to remember that the data are collected not for research. They're collected for care. They're collected for payment, either for the doctor or the hospital, but definitely not for care. And as a result, what gets collected is slightly different to what would be collected in a really, really well organized research study. So how do we go from the raw building blocks, like a ton of Lego bricks, to sort of the Millennium Falcon? And that process, by going from raw data points, to an algorithm that can then be statistically analyzed, is called phenotyping. And phenotyping is what I and sort of the people that I work with spend most of our time doing. There are broadly two kind of ways that you can phenotype electronic healthcare records. One is what I've called shallow complexity. So you basically create algorithms that identify who has a disease. Given someone's electronic health record, does this person have glioblastoma? Yes or no. Does this person have colorectal cancer? Yes or no. And then there's also more deep complexity algorithms that deal with recurrent events, that deal with probabilistic, and what is the probability of someone having a particular type of disease. And there are more machine learning kind of approaches that are used in that area. But that's kind of the main, sort of the two main families of approaches that we use. So I don't know if you've ever seen this XKCD comic that kind of floats around from time to time, and it sort of talks about modern digital infrastructure and things that are like developed by very big kind of tech companies and things like that. And then it kind of says that, this is all kind of, put into place by a project that some guy in Nebraska has been maintaining and has open sourced it on GitHub, and they just reuse that. So I kind of see a lot of the things that I do as like this kind of providing these tools and the scaffolding for then, high throughput drug discovery and then epidemiology and, genome-wide association studies and clinical trials to happen. So phenotyping kind of is one of the fundamental kind of things you need in order to define the disease you want to study or to identify who has what condition when you're trying to develop a pipeline. And that's kind of something that I'll try and show through all these different sort of presentations. But it's something that I kind of take pride in, that this is sort of it's the tools and the methods that get reused over and over again and lead to sort of even better things that I alone as a computer scientist would never be able to do, because you need sort of this multidisciplinary research with clinicians and kind of policymakers. So I'll talk about kind of the journey of all the things that I've worked with since I joined UCL and how we went from studying a handful of diseases at a time to studying like the cosmos of diseases, so all diseases. So this is one of the first algorithms that we created. It's to define a particular type of cardiac arrhythmia. It's called atrial fibrillation. And it basically takes all these building blocks from all the sort of different data sources that we had linked back then and identifies who has this disease, this arrhythmia, who doesn't. And we also did some clever stuff to try and identify who possibly has atrial fibrillation, even though they don't have a diagnosis. So who has similar patterns in terms of therapies or procedures that indicate that they might have that, but there's no formal diagnosis. And then we used that, and we developed all these other algorithms to define these 12 cardiovascular disease cardiovascular disease endpoints. So we established the platform in 2011 called Caliber, which back then was sort of the largest cohort study that you could study in the UK. Then we linked data from family doctors to hospital to death records and cardiac registries in order to study cardiovascular disease at scale. And we did really, really simple analyses looking at recorded blood pressure and how does recorded blood pressure, systolic and diastolic, increase or decrease your risk of these 12 cardiovascular diseases. And why that was sort of really, really important is that traditionally, because of the small data sizes, these diseases were usually grouped together into larger groups. And you were unable to see that actually systolic and diastolic blood pressure have slightly different effects on your risk of these diseases, and also that the risk between and across these diseases is actually heterogeneous rather than homogeneous. So there are significant differences in how systolic and diastolic blood pressure influences your risk of these diseases. And these are sort of endpoints that traditionally don't end up in trials. So this was kind of our first landmark paper. This is where the data was stored when I first joined. So I had to spend a very long time in a very cold room to set up all these sort of like things from scratch. And I still remember when I joined UCL, it was kind of, everything was in Windows and it was 32 bits. And we were sort of like emailing IT and we're saying, hey, we want to load this file with, X million records. And they're saying, well, you can only have 2 gigabytes of RAM because Windows doesn't support it. So we had to do a lot of the heavy lifting. And that's us celebrating our first kind of paper back then with the forest plots that I showed you. And I used to look much, much better back then. And then how do we scale that up? So we went from studying a handful of diseases, 12 diseases, to studying many common diseases at a time. And this is particularly important because at least in the UK, multimorbidity is rising. So people live longer, but their quality of life is not getting any better. And as a result, they develop more and more conditions, chronic conditions, as they're called, multiple long-term chronic conditions. And by the age of 85, almost 81% of individuals have 5 or more chronic conditions that they live with. And on the right, you can see that there's a clear socioeconomic gradient between the least deprived and the most deprived areas of how these manifest. So multimorbidity and polypharmacy, which is the use of multiple medications at the same time, are also increasing. And it's also important to find how do patients move, how do these diseases manifest across the different healthcare settings. So again, in the UK, which is unique in how the healthcare system is created, there's a lot of diseases that only get handled by your family doctors, things like type 2 diabetes or hypertension. So usually it gets managed and treated by your family doctor, and then more severe manifestations, you end up in hospital and you go back and forth. So using electronic health records linked enables you to study this longitudinal pathway of disease. So this is work from Valerie Kwan, which sought to answer this really, really simple and elegant question: who gets what disease and when? So we were able to link data in now 4 million patients, a step up from how we were in 1.2 initially. We picked 50 diseases that are associated with a high healthcare utilization and burden, working with clinicians. And we basically identified which diseases occur at which decade of life and how do these diseases differ by age, sex, and ethnicity. And again, we used phenotyping algorithms to define who has what disease and when. So we were able to kind of show, for example, that infections are extremely common in the first decade of life and then in sort of the 80 years or more. And other diseases like cardiovascular disease are more common in the sort of the first manifest when you're 60 to 70, mental health when you're 30 to 39. So this is the median age at first diagnosis, and you can sort of see the gradient that you would expect, that cardiovascular diseases like 70s, cancers are around 68, and then things kind of go sort of slightly earlier in life, with mental health being always or almost always one of the first manifestations you get. Then we wanted to look at which diseases occur with each other, and are there any differences between men and women. And this is the co-occurrence sort of graph of asthma. And these are all the different conditions that occur with asthma within this cohort, non-randomly adjusted for multiple, sort of with multiple comparisons. And we can sort of see that even the fact that the same diseases occur in both men and women, the magnitude of the association is different. And also by definition, women have substantially more conditions that are only conditions that women get that are associated with asthma, such as PCOS and dysmenorrhea and menorrhagia and others. And that was sort of really, really interesting, because we could then look at the differences in ethnicity and again identify how these comorbidity networks vary by ethnicity. And the UK is highly multi-ethnic and multicultural. And we sort of, one of the things that we found was that atrial fibrillation was sort of highly associated with primary breast cancer, which is kind of in the Black population is actually very common as a symptom, and indicates severe aggressiveness of the breast cancer. And this is the whole map of multimorbidity in all patients, and it's kind of reassuring to see that there's this cardiometabolic cluster, that things that are sort of clustered together, and all these diseases that are really, really close together means that they occur very, very frequently in individuals, non-randomly. We're also interested in aging, so a subsequent study looked at clustering these diseases and identifying which diseases have an increased risk of occurring based on aging. And we used hierarchical clustering, and we identified these 4 clusters. First one was enriched for dementia, second one is cardiovascular, third one is cancers, and the fourth one is digestive. And you can see from sort of the line, they all have a sort of an increase as older as you get. Aging is by far, at least again, in the majority of diseases, one of the most kind of strongly associated sort of risk factors that is obviously non-modifiable. And this is, again, the median age of onset across these 15 broad categories, and dementia is definitely one of the top ones. And it's interesting that the median age of them in that cluster is 82, because we know that dementia has a huge prodromal syndrome, prodromal kind of period, at least in the UK, that it takes like almost 10 years to get diagnosed, to get a firm diagnosis of dementia. So that has kind of implications of how dementia gets diagnosed and managed. And this is the cluster composition. I won't sort of dwell on that for time. This is a really good example of that XKCD comic that I showed you. So this is work from Julia Carrasco that she basically took the phenotyping definitions for all these conditions and tried to identify which plasma proteins can be used to predict the risk of getting these diseases. So she used our definitions and run a lot of models to find out which plasma proteins can increase the risk of prediction. And she found, I don't have the actual number here, she found close to hundreds of individual plasma proteins that can be used to increase the accuracy of predicting within a clinical setting with the minimal data, the risk of getting a disease. And that sort of subset of plasma proteins can potentially be used to develop a risk score that can be run in clinics. And this is an example that, if I'm honest, I wouldn't, this is not my area and I wouldn't be able to do it. This is only possible from working with other people across bioinformatics and clinical bioinformatics that it's possible to do it. And then the last section of my talk is about studying all diseases and scaling everything that we've done to span the entire phenome and study all diseases at the same time. And then it's with all the interesting connotations of what is actually a disease and how many diseases are there, which I've chosen to ignore to dwell on at this presentation. During the COVID pandemic, something happened in the UK that was sort of a monumental change. So if you've sort of looked at the slides that I showed before, I was talking about cohort sizes of 1 million, 4 million, 6 million. During the pandemic, the Secretary of State wrote a letter to the National Health Service and said, stop the excuses, make all data available for health research for COVID because this is a national emergency. And literally overnight, we had access to 56 million patients worth of information and their entire medical history for research into COVID. Obviously, very, very strict securities, remote access only, and lots of stuff going in that. But we were able to sort of do research using the entire thing. And this is a sort of an early paper that we did on COVID trajectories with Johan and Chris, in looking just at the very simple question is how is COVID-19 recorded across different sources? And they showed that actually no single source is complete. And I'm sure in Austria it was the same as in every other country where the numbers were always debated and is it the true number of cases? What is the mortality across different groups? So we showed that it's actually quite diverse and each source captures its own unique portion of positive COVID-19 infections. And equally, the trajectories between the different states varied between waves. So we were able to show that in the first wave, which was by definition the most severe with the alpha variant, the transition from, let's say, uninfected to death was much, much shorter than in the second wave, that we had sort of better control of sort of pandemic preparedness. And this is all, but before vaccination. So this is again using the national health records data. This is work by Stephen that was sort of looking at under-vaccination and showing that actually under-vaccination, which was a big problem in the UK, because by 2022, roughly 50% of the population was under-vaccinated. Under-vaccinated doesn't necessarily mean unvaccinated. It means that they might be unvaccinated or not have all the required doses for their sort of risk profile. And sort of you can see a gradient of age and the increased risk of under-vaccination. And we also looked at, again, with Johan, at the prevalence and demographics of rare diseases. And when you have, again, data of that scale of the entire population, we compared the prevalence of diseases, or how often diseases occur in the national data set compared to what we thought we knew based on estimates derived from different sources. And we showed that almost all estimates actually underrepresented the true prevalence of many, many of these rare diseases. So it turns out that they're much more common in the healthcare system, at least in the UK, than we thought. And probably that's safe to say they're much more common in most other healthcare systems as well. And again, an analysis that's only possible when you have data at that scale. We also did similar analyses for cardiovascular diseases and like trying to show the value of using the data and identifying trends and changes. And this is a recent study looking at the trends of cardiovascular disease and looking at there was a gap, there was a decrease in most diseases because of the pandemic and the shielding lists and the quarantines. And then most diseases recovered, but other diseases persisted to be decreased. Other diseases like myocarditis increased. So that it shows you the power of using the data at a national scale. Unfortunately, the reality is that all of these permissions have been backtracked now. So we are losing access to this data set. We know it's possible to do great things, but it's very, very challenging to use it for non-COVID research. So my final slides are this notion of lumpers versus splitters and kind of treating diseases as homogeneous versus heterogeneous. And I like this sort of phylogenetic tree. This is from the sunflower, and it turns out that the sunflower has more than 26,000 different species in its family. So there are more than 26 variations or permutations of the sunflower. Yet, philosophically, we choose to call type 2 diabetes as one thing, type 2 diabetes. And everybody on the world that has type 2 diabetes gets this one label that makes life easier, but might not reflect the underlying heterogeneity. So the way that we've tried to approach it is to take patients with a particular disease, extract a lot of clinical features, and then use clustering and supervised learning to identify clusters that are enriched for different diseases. So this is work that Ami did that led identifying clusters in 300,000 patients with heart failure. And we've identified these 5 different clusters. But more importantly, we were then able to look at the genetic data behind these clusters and find that they were enriched for particular genetic mutations, for particular SNPs that reflected kind of the underlying biological meaning of these clusters. And that's kind of the same approach that we followed for COPD as well. And we were able to show that patients kind of very neatly cluster in 5 distinct kind of clusters. This is work by Maria. And importantly, these clusters have significantly different healthcare utilization patterns. So we were able to show, for example, that the cluster with anxiety and depression, which was mostly younger females, had highly increased rates of consultations with the healthcare system. So you could imagine interventions into that particular kind of management of those groups and of the patients. And then similar thing with COPD and other respiratory disease subtypes, where Maria basically showed that these conditions are highly overlapping and they don't actually fit in this traditional label of calling something COPD only or asthma only, and that these actually very different sort of physiologically. So all of what I sort of described is only done by kind of working with others, and I've been sort of very privileged to have had excellent mentors. And I use the word mentors in a very loose way. I mean, some of these people might not even know that I consider them mentors, but these are like all individuals that I've worked with a lot and shaped a lot of how I think about things. And then, countless of colleagues throughout the UK and elsewhere that have made this possible. And then also, we're very grateful for a lot of funding that we've received over the years to make a lot of this research real. And thank you very much.
I'm pretty sure that there might be already some questions regarding what Spiros is working on, but also regarding what does this mean, how does the future look like, what can we learn from that, etc. I would ask you to hold back a bit more, and even you joining online, so that we can also listen to our complementary second interdisciplinary talk. And for that, again, I would invite Stephanie to give some welcoming and introductory words to Kristin.
Thank you. Well, so Spiros now came to Linz to revolutionize the Austrian healthcare system so that we also will be able to finally get our data, I hope. Because I think there the UK is far advanced to what we have here so far. Maybe one thing more on Spiros is that he is still keeping his group at UCL. So he's building up a group here as well as there. So we have the benefit of still working with the London colleagues over there. And so I think this is a great opportunity also for students to come and visit and go to the other location, so to say. Okay, so with this, I want to come to Kristin Reiche. She joined us this week, I think, right? So, or last week? Last week. So very, very new here in Linz. So she comes from us from Leipzig, where she also studied computer science already with the minor in medical informatics, and then also studying, doing her PhD in bioinformatics. So a very similar trajectory as Spiros. And then after receiving her PhD at the University of Leipzig, she did a number of different postdocs positions. Fraunhofer, Helmholtz, these are these more applied science organizations in Germany, like here the Comet centers, these are comparable. And then developed into a group lead at Fraunhofer, became department head at the University of Leipzig again, and through different things, finally arrived at ITU, where she now will build up her group in computational medicine, and you will now explain to us exactly what's similar to what Spiros is doing and what's different. The floor is yours.
Thank you very much. So thank you, Stephanie, for the very nice introduction. So I would like to present to you today a very central question actually to my research, which is, so I'm here at ITU as professor of digital molecular medicine, meaning a central question for me is it actually possible to digitalize molecular medicine, or is this actually a moonshot project? And today I want to address that in my talk with three topics. So first of all, I motivate why molecular medicine is important, why it does matter. Beside all the electronic health record data we actually have available in computational medicine, still the question is, what does molecular medicine contribute in addition? Then I will outline what I see as potential barriers, or actually, or not just potential, but real world barriers for implementing or making molecular medicine available for most of us. Because this is still not possible or not achieved. But I do have some concepts where we find a solution, and hopefully here together with at ITU in Austria, in the upper Austria part. So we all actually hope that for our complete lifetime we stay healthy. Okay, so this is an achieved or a status we would like to achieve, but it could be the case that for some, or we often also get a disease, and one major or one still devastating disease is of course cancer. Still, it can be well diagnosed and also to some extent already quite well treated in countries which are high developed, meaning that we do have here, of course, also already some patients which survive their cancer. Still, it's a disease or a type of disease actually which may face us, half of us sitting in the room at once in our lifetime. It could be the case that we get diagnosed with cancer, and it of course also affects female and male. It's just a disease which we need to understand in a much better way in order to be able to treat it. So which factors drive actually cancer? So it's genetics to some extent, but also lifestyle. But the point I want to make here is that everything what actually is related to cancer is that we observe that different cell subtypes actually change their status from a normal, from a healthy status to a disease status, to a cancerous status, and the process leading to these cancer cells is called oncogenesis. And it's in well-recognized publications by Hanahan and Weinberg described as different hallmarks of cancer. And these factors which drive which actually define the process, how a normal cell is actually evolving to a cancer cell, is here described in the hallmarks of cancer, that we have many molecular and also cellular factors which lead that a cell which is not presenting a specific molecule on the surface anymore and cannot be recognized by the immune system anymore, in order that this cell then proliferates hugely, and the cancer can further evolve in the body. So we know that if we face types of diseases like different cancer diseases, that cell biology or molecular biology matters. And now is the question, how can we actually assess the status of different cells in order to better understand what actually triggers this process of oncogenesis. So you see here 3 cell types just provided in different colors. Each of these cells, even though they have a different function in the body, could be that the red cell is just a skin cell, and the green cell is a cell in the brain, that all these cells actually have the same genome, but specific regions in the genomes, the so-called genes, are transcribed to the transcriptome, which is unique and described for each of these different cell types. And following the central dogma of cell biology, we then actually observe that this transcriptome is translated to the proteome, and all this together, all these molecules, these transcripts, these RNA molecules, together with the different proteins, they actually define then the phenotype for a specific cell, and in combination for the whole body actually also define the phenotype for a patient. And also with that, if we know these particular data sets, if we know how the genome looks like in a particular cell subtype, in the organ, in a specific tissue, at a specific time point of a patient, then we can much better understand which type of treatment will actually provide the best response to a particular cancer type. And since technology advances, we now have the chance to use the so-called next generation sequencing technologies. Here just, I give just a very short overview, because we have many different devices available, but I just want to show that if we use next generation sequencing, we are actually able for different cells to characterize the genome or the RNA molecules, the transcriptome, and to some extent also the proteome, the proteins. Meaning, if we use next generation sequencing, we can actually characterize the different cells. So it's quite important to understand which type of data is generated by these technologies. And here I give you an example. So we do have several categories. For example, one is so-called bulk sequencing. And here you see the different circles just highlighted in gray. They depict now cells originating from the same tissue, but usually these are different cells. But when we use technologies from the bulk sequencing category, we cannot discriminate which of the genes which we measure actually is assigned to which cell. So just to give you a picture, in the end, if you do next generation sequencing, you just receive a mixture of a smoothie with. You are not able to actually identify the ingredients of this smoothie. But if you conduct single cell sequencing, which is another type of technology, also using next generation devices, you are now suddenly able to tell which ingredients the smoothie had, which is a huge advancement of information we receive. And just for a couple of years now, 3, 4, or 5, or just 1 year actually, we are now also able on an intercellular scale to assess the gene activity and the transcriptome activity in a spatially resolved manner, meaning that we do not just know to which gene, to which cell a particular gene which is highly expressed is or originates from, but we also know which of the cells actually are in close proximity to each other, which again gives us a much more informative data source. And here, just to give you an example from my lab, so we conduct single-cell sequencing. Here on the left side for single-cell sequencing data, you see T cells which have been characterized by single-cell sequencing, and we here can highlight the different clusters, calling which of the cluster is is related to CD4 plus T cell or CD8 plus T cells. And to understand these T cells and the different molecular patterns of the T cells is quite important to also understand how our immune system is able to fight cancer cells. But in another different picture on the left side, you see now also a data set which has been generated in my lab, where we used Visium HD high-definition spatial transcriptomic device, where now suddenly we are able to pin down the position of a particular T cell within a tissue. Here, a melanoma tissue, so it's a skin cancer tissue slide, and here we can now understand what molecular status of a particular cell is related, or how does the molecular status of a particular cell type change independent or dependent of the neighboring cells. So, and with that, I want now to define what we see as molecular medicine. So molecular medicine is an application of single cell or genomics or epigenomics, transcriptomics or proteomics. All of these layers can mostly assess with next generation sequencing. And we use this different set of data types in order to understand, diagnose, and also in the future to treat disease. And this is not only just the future to treat diseases with cells, so with different genetically modified cells, but one possibility is here the so-called CAR T cell treatment. And this is a quite successful treatment for some blood cancer types, where from a patient, the cells, so the T lymphocytes of the patient are taken and then in a GMP facility manufactured and genetically modified in such a sense that the T cell then carries a chimeric antigen receptor, so a CAR, so it's not the CAR where we drive in, it's a chimeric antigen receptor, which is put on the surface. And this receptor is able to identify the surface marker, which is usually presented on the cancer cell. And if these CAR T cells will be given back to the patient, if he or she receives an infusion product, then these CAR T cells are able to modulate the immune system in such a way that the immune system fight the specific cancer cells. And this treatment is quite successful. So here you see Emily Whitehead. She is one of the very first, or she's one of the first humans actually who received CAR T cell treatment. And she is now, after many years, she now says for herself that she is cured by, from cancer because she had access to this innovative type of treatment. But if we provide these cellular immunotherapies to patients, we face following problems, which is that compared to conventional drugs, we now give to the patient not just a cocktail of different pills, but we provide the patient with a treatment which covers and combines living cells. So it's a biological system we give to the patient as treatment, meaning here we do not have just the impact of this molecular or the cellular status of the cancer tissue, but we also need to better understand the CAR T-cell product in order to see who is responding well or who may face actually, or who may have a risk of to face toxicities. So to summarize that, with the advancement of engineered adoptive cellular immunotherapies, this is it's a big hope for patients, but we do have patient and in addition product-specific treatment responses and side effects, and because of that, computational medicine, also bioinformatics, has to take this into account and to drive the field forward with different tools. So, but we see barriers for implementing molecular medicine for most of us. So, and these barriers are, I will just tell you about that more in a couple of seconds. But for now, we do have from the technologies which are available, we do have a huge innovation on the research part, meaning that more or less every couple of years, Nature Methods actually assigns one of these multi-omics technologies as the methods of the year, starting with sequencing, next generation sequencing, then single cell sequencing, and then spatial transcriptomics, and so on and so on. But as long as, of course, it's important for using these technologies and devices for research questions, it's also important to find ways to implement it into clinical routine. And why this is not just a future or a story which I see in the future is because Illumina already provided a device which is CE certified, so compliant to in vitro diagnostic regulation, which can be used as an in vitro diagnostic product in clinical routine. And now, just last year, Singlon is a company in China, but also in Germany and in Europe, provide a first-class medical device for automated single-cell analysis. Meaning that in the next years to come, I also see that single-cell devices and single-cell sequencing will be not just used within the research setting, but will also find their way through clinical application. But if we conduct literature research and to understand in how far these technologies, because they are quite advanced and they are not available in all the different labs, in how far they are available, or in how far, or if they are actually accessible to everybody who is in need. I can tell you something about that, which is that the European Cancer Organization Action Report of 2022 outlined that these technologies are important because they provide us a deep insight into the different potential molecules and biomarkers which are important in order to decide which type of treatment can be given to a particular patient. But we do also face problems in providing access to these technologies because they are expensive, because mostly they, for example, molecular tumor boards are only provided at university hospitals and urban areas are often left behind. So we do have white spots, meaning that these technologies are not available to all of us. So what are the barriers? The barriers are that they are quite expensive to use. And the turnaround time, so meaning when a sample is given to a diagnostic lab until the sequencing is conducted and data analysis is done, it's quite long. So if diagnosis has to be done in a very fast way, then we face a problem here. Then also the socioeconomic status is still a limitation of access to this type of diagnostics. And we do also have regulatory constraints because they are quite advanced technologies and not all of those are yet available as an in vitro diagnostic device. But since we know it's quite important to actually use also these technologies in order to characterize tissues, biopsies, and to define the different potential biomarkers which can be used in order to decide which type of treatment fits the best for a particular patient, we need to find solutions. And here I just list several ones. And in my, due to time reasons, I will just concentrate on 3 topics for the last part of my talk. But it's important here that we have different, different building blocks we have to concentrate on. Like, for example, it's also possible or important for single-cell sequencing, data analysis pipelines, that these pipelines are not just able or just available for research operations, but also for clinical applications. And for that, we also require full interoperability of the data sets and the different communications between the different systems. So we do have many different building blocks we can address by digital molecular medicine. And this is something I'm really eager to follow up here with ITU. But for now, just concentrate on 3 particular topics, which is learning from real-world data. As Spiros already nicely outlined, real-world data is available data source and also for molecular medicine. Then interdisciplinarity, of course, is very important. And also from the very beginning, compliance by design for software, which can then later be used as a decision support tool, either in a medical device or an in vitro diagnostic device. And this brings me now to the last part of my talk, which is to give you some ideas how we in the past implemented some of these solutions in order to pave the way for advanced diagnostics tools in molecular medicine to be used in clinical decision making, which is that our aim is in my lab that we make the best out of multi-omics technologies. So we want to make data sets like we generate via genomic sequencing or transcriptome sequencing or spatial or single cell sequencing as close as possible available for the bedside, so for the patient. And what we implemented here as a research team is that besides having a lab which generates omics data, we can also provide all the bioinformatic and data analysis pipelines, but do that for some relevant projects also under a certified quality management system. And in addition, also we implemented the software lifecycle according to a medical device and in vitro diagnostic regulation. Meaning that if we start a collaboration with partners and see that a particular analysis of a data set may end up in a very important biomarker signature, which will help patients, we can directly start to implement the software implementation and documentation according to IEC 62304, which describes the software lifecycle for medical and in vitro diagnostic devices, so for software in medical or in vitro diagnostic devices. And we have done that in the past for developing transcriptome signatures for diagnosing and prognosing prostate cancer. And for these novel adoptive cellular immunotherapies for the CAR T cells, we use also cohorts from from hospitals in order to learn from real-world data. And here we follow the same strategy that we form in my department an interdisciplinary lab of wet lab scientists as well as bioinformaticians, so tri-lab scientists, in order to assess the samples under all the certifications as it's required to make the best of the data, and also to document what we have done in such a way that it's possible to develop a biomarker signature later on, which then can be certified as an in vitro diagnostic device. And here I just want to show you some results. So for CAR T cells which have been given to patients which suffered from multiple myeloma, we were able to receive on a longitudinal scale different blood samples, did single cell sequencing, and here we were able to identify different cell subpatterns of different CAR T cell products which are related to a good or to a bad response of the patients, as well as different cell subpopulations. We could show that they are related either to long-term survival or not such a very good response to CAR T treatment. So these are just examples that also with single cell sequencing on the molecular level, we see associations to response to the CAR T cell treatment, which then gives us advices how to innovate and how to change preclinical development of future CAR T cell treatments. And for another study, we also use single cell and spatial transcriptomics and the according bioinformatics and machine learning methods in order to assess for a particular patient who actually had a severe side effect of CAR T cell treatment. And at this time, this eccentric question was, is this side effect actually, did it occur due to the CAR T cell treatment, or did the patient has a genetic predisposition? And we could identify with these technologies that there was a genetic predisposition, that some of his T cells, then after they received a CAR construct, actually evolved to cancerous T cells, and which is really a severe side effect of CAR T cell treatment. But it was a very important study for the field in order to understand why these side effects actually occur. And this has been published in several publications in the last couple of years. And we now combine that into a EU-funded project, which is called Certainty, which I coordinate, where we aim at providing the very first prototype of a virtual human twin, of a digital twin, of patients which are eligible for CAR T cell treatment. And since virtual human twins is an emerging concept in personalized medicine, we still have not included molecular data well in these type of models. And here in Certainty, we aim also integrating cellular, intercellular, and molecular data in virtual human twin models. And how do we do that? Just for an example, here we partnered up with UMC Utrecht and Institut Curie, and here we use a Boolean. So the Institut Curie, Laurence Calzone, is an expert in modeling single cells with Boolean networks, and we use also data from longitudinal single cell sequencing data from patients, as well as in vitro models. So we can put this data, these samples which we received from patients also into models which can then be studied in the wet lab, and how far this particular sample is actually responding quite well to a particular treatment. So we combine these different data sets in order then to provide personalized models, in silico models, for patients who received CAR T cell treatment. So to sum up, we have a very novel type of treatment available, which is engineered adoptive cellular immunotherapy. And one of example of those is our so-called CAR T cells. But since here we actually provide to the patient who is the living system a biological system, we have additional factors we need to integrate in order to well understand who is responding well and who is not responding well, which is the infusion product, so the cells which are given to the patient. And because of that, we need innovation in molecular medicine, which covers that we also require validated bioinformatic pipelines, inclusion of real-world data, but also full interoperability of digital solutions, also in molecular medicine, and not just in medical solutions where we concentrate or use data from electronic health records. How do we want to implement that into the CAR-T product lifecycle? So if a CAR-T treatment has been approved by the European Medical Agency, then it enters the market, and it has to be also then followed up after approval, so in the post-market phase. But with the projects we conduct in my lab, we use learnings from longitudinal single-cell multiomics data, from the real-world data, which I just described. And then we can use these single-cell data and the learnings, which cell status and which molecular patterns are related to a good or a bad response to this type of treatment. We can use this information in order to edit also in a genetically way the cell biology for future CAR-T treatments, so we can improve future cellular immunotherapies. Then if we partner up with different disciplines, we are able to establish lab in the loops, where we use also data from real-world examples, and in order to test that again in a wet lab setting. And then what the hypothesis which has been tested in the wet lab can then again be transferred back to the in silico model. And of course, it's important here to also in the preclinical development of new CAR-T products to integrate learnings from real-world data. And since the FDA now also requires randomized control trials also for this novel type of treatment, we also need new data and also in silico data in order to prove superiority over existing CAR treatments. And here virtual patient twins can support the market approval of new CAR T-cell treatments. And since we provide patients with a treatment which consists of cells, for some of these patients we observed long-time persistence of these cells in the body. And because of that, we also need to provide support from the molecular medical point of view for lifelong monitoring. Patients also ask for shared decision-making. So they want to be informed prior they meet with a clinician in order to decide, is this treatment the option I should go for, or should I choose a different option? And again, if we learn from real-world data for future CAR-T products, we find a way how to feed information from the bedside back to the bench, and this can then be used to improve future cellular immunotherapies. And in order to achieve that, I want here at ITU implement the following strategy. So first of all, we also need to improve the digital competencies of caregivers and patients. And this can be done, for example, by providing an annex to patient consent forms in order to also explain quite well what does it mean if genetic data or single-cell data or spatial transcript data is actually generated from your sample. And here we need also, we need to aim at literacy of the patients, so they need to understand what does molecular biomedicine or medicine actually mean. Then also this can be conducted by information sessions or cafes with patients. And we also need to find new ways and concepts for reporting results of molecular medical devices. And we also need to implement the digital process for molecular medicine in outpatient and inpatient care. Here we can follow up all the in vitro diagnostic regulations, and why we conduct software implementation according and compliant to the in vitro diagnostic regulations, so to the software lifecycle according to 62304. And this has to be not covered for the Illumina devices, but also for 10x or third nanopore, third generation sequencing devices. So there's many things are ahead of us. And we also need to further integrate molecular data into virtual patient twins. And in the end, we also need to provide that on the European scale to different stakeholders. So we are in contact with the European Virtual Human Twin Initiative in order to integrate also molecular data into these models. And this has also to be aligned with the European health data space. But all this is not possible without interdisciplinary collaboration. And here, this is not just a picture which I just made. This is really providing an information of the background of the different disciplines I used to collaborate in the past, not just within the Certainty project, but also with different projects. So we need interdisciplinarity also for molecular medicine in order to make molecular medicine available for most of us, at least to the ones who are at need. And because of that, I would like again to highlight that beside the many different data types we use in medical devices already, we should not forget about the omics layer. And please don't forget that omics is actually not just one bubble, one circle there. It actually spans or covers many different data types, which all have to be integrated in medical devices or in vitro diagnostic devices as well. Because for patients like Emily Whitehead, it's a big hope if we make molecular medicine available for most of us. And with that, I'd like to thank all my collaborators and also the patients and funders, and thank you for your for listening, even though it's 4 minutes longer than expected.
Well, thanks a lot, Kristin. And with that, I will also re-invite Spiros on the stage. And then the floor is open to the first questions. We have like a microphone, so also that the online participants can listen to a question. Who wants to start with the first question? Then I'll give it to Stephanie.
Hi, my first question goes to Spiros. I was wondering, how does I don't know, if you had time already to really look at the Austrian health system and the data, how does that differ from UK?
I've had time. I have met with various people that have explained it. I think it's different from the UK in terms of, I mean, first of all, Austria is significantly more conservative about the use of health data for research. Similar to the UK, there's no central data repository, and it sits within different insurers. So we don't have insurers in the UK, but it's kind of fragmented. But there are really cool studies that have come out of Austria, especially from the Vienna Complexity Hub on multimorbidity that have used data similar to what I have from hospitals. So the data are there. It's just very difficult to access. So one of my plans is to try and use publicly available data and organize some sort of hackathon or some event to show the value of using data to answer similar questions in a much simpler way, and then hopefully build some collaborations. Yeah, but I think it's going to be difficult, but we'll see.
Yeah, maybe it's more an indirect question, because I was wondering where are, for example, the rheumatic disease, because, I mean, you taught a lot about cancer, both of you, and also, especially you, about old people. But, well, I was working in a hospital, and in fact, many of the old people simply don't want anymore. And additionally, in a hospital, in medicine, you make a difference between when what a disease does and what the body itself does. And there are many cases when a disease is already over, but the body is already simply done or too old, and then the people die. And on the other hand, you have rheumatic disease, where often young people are affected, and they simply don't reach that high age. So there are maybe not that much, but yeah, I was simply wondering if you know anything about that group, because it also didn't shine up at your slides. Yeah, please.
Of course, so for autoimmune diseases in general, now these novel type of treatments, these CAR-T cells are also available to some extent. It's still just for some cases, but it's also here a good hope. But if we put that into in the frame of my talk, actually, then it actually gives us not just a small cohort of particular type of cancer patients, but to just a larger number of patients receiving these living drugs, putting pressure again on the system that molecular medicine and in vitro diagnostics should be quite well advanced and digitally and integrated into the care, into the process. So yes, so I didn't talk about it, but I'm aware that at least for these treatments, it's also an option for these patients.
Yeah, I mean, I also didn't show it. Part of those 300 diseases that we did the life map was rheumatoid arthritis and lupus and other autoimmune diseases. But I think to your point is, so this is a limitation of the data. So compared to the rich molecular data, we get the labels that the healthcare system uses for payments. So we know someone has a diagnosis of rheumatoid arthritis, but we have little information on how that progresses or how that expresses or all the manifestations. So we know they have it, we know if they have a particular drug for it, but that's about it. So I think it's a limitation of the data that you need to combine with molecular data to really understand.
Are you really going towards, well, the center or I don't know, the hospitals that have these patients to get your data, or how do you get it? I mean,
so you can do both in the UK. It's easier to go for the central data collection approach, which is a central database that the healthcare system has for payments to hospitals. So every hospital submits ICD-10 codes for all the patients they've treated, and then they get paid based on those patients and the severity of the condition. So we use that database for a lot of research, or the data that they present with at the family doctor. You can definitely go to individual hospitals, but that's much, much harder to do for access and time.
I follow the hard path. So I really go to individual hospitals and reach out to the clinicians and collaborate with those which actually follow a very smart biobanking strategy already, because everything what I just presented today would not have been possible if not a particular nurse and clinician actually took care of taking the samples and store it in a very well-documented biobank. So yes, both is possible, but mostly for molecular data, you need to collaborate with the particular hospitals. Yeah.
Thank you. Thank you for the interesting lecture. Considering the hindrances that are in the access to data, what is the role of synthetic data play in research?
Obviously, hugely important. I think there are various layers of synthetic data. So the simplest would be something that we often sort of use, is that something that's semantically the same as the real thing. So the column names are the same and the data types, but you can't really do any analysis because the values are wrong and there's no correlations to be found there. So that's useful for developing pipelines when the code sits in a remote sort of environment and you can develop it locally and then push it. And then there's synthetic data that actually is mimicking the real thing that you can actually use for teaching or for running analyses that kind of replicate the real thing. But the challenge, at least in my experience, is that when such a real-ish synthetic data set is created, it's usually created through some sort of algorithm that's applied on the real data and then is abstracted and derived. And people end up using it in the same way as the real data set in terms of information governance. So, and they say you have to store the synthetic data set in the same way that you store the real data. And then that kind of defeats the purpose, because if you're going to have to, you might as well use the real data. So I think it's kind of the balance between, but it's something definitely that's very useful, at least for the stuff that we do.
Yeah, because it can be used to train the models. If there is some kind of algorithm that produces this synthetic data, and then the algorithm can be trained based on this data.
Yeah, so we don't do that. So we only use the synthetic data to develop the pipelines. I know what you mean. I think, yeah, our synthetic data is just data that looks real, that you can develop your code on, but it doesn't have any meaning. So you can't really train or infer anything, unfortunately.
Thank you very much as well. My question goes to Kristin Reiche, which would be, I would be very much interested to hear more about what is possible with a digital twin of the human body. This is something that could, of course, have a lot of impact for a lot of diseases. So I would like to hear what is the current state that we're in there.
Digital twin modeling is actually that there's quite successful models available for particular organs like the heart, and which is also used for teaching, or also actually preparing a particular surgery, for example. But to aim for the virtual human twin, the whole body virtual human twin, this is really an international moonshot project. So even the one in Europe, so the key opinion leader here, Lisbeth Geries, actually had led the EDIT consortium, which wrote down the roadmap, what we need to do in order to get towards a virtual human twin, covering the whole body. And yeah, she herself tells everybody, we need to do the different steps, but we won't reach it now in the next couple of years.
Maybe a very quick yes/no question in that regard. Do we have enough understanding to do this right now? Okay.
No, because as you just saw, so biology is the biology is complex, and as you just seen, we are not at the stage that we, at each of the different disease status, we have data available on the cellular and on the molecular scale as well, which is required also to model a whole body virtual human twin.
Thank you.
Yeah.
Oh, thanks for your talks. It was really interesting. My question is to Spiros. Like in your slides, you also mentioned mental health and phenotypes of mental health, like how to, like, what are the phenotypes that lead to a mental health diagnosis. So can you, like, I don't understand how, like, is in the UK, is the mental health system integrated with the primary and the hospital system, and is the data available similarly to how other data is available for doing that analysis?
For mental health specifically, yeah, it's different in every country. In the UK, again, everybody's registered with their family doctor, and the family doctor is always the first point of care, and so it's safe to assume that when you see a mental health diagnosis in a hospitalized patient, it's a more severe presentation than something that's managed only in primary care. So you can then see the prescriptions, and you can see sort of various sort of healthcare utilization patterns, but the data is not as, so there's two limitations. One is that, especially in mental health, the majority of data points are created using text. So the doctors write a lot of text reports that are almost never available for research because they're highly sensitive. So we don't have access to that. And the second thing is that it's such a diverse field that it's really hard to kind of get rich data. So again, that's where you need to go to specialized cohorts like the UK Biobank or future, that collect a lot of repeat measurements specifically to mental health that you can use. So routine data alone is probably not enough for a lot of the questions, I think.
So, like, part of your work, does it also involve, like, collecting as in phenotype is here based on interactions with the primary health centers and hospitals, right? Is there also efforts to look at phenotypes as human to environment interaction, other sorts of interaction, and collecting data?
Yeah, we do that. I mean, we use interactions as a proxy for, let's say, severity. So if we define two groups of patients that we think are different presentations of the disease, and one goes to interacts with the healthcare system 6 times more than the other, we can then infer that they have an increased healthcare utilization and they're more severe than the other. So we do that kind of analysis, but if that's what you mean.
What I was referring to was also using other data sources to build this phenotype graph, but yeah, thanks. Thank you.
Follow-up question to the digital twin, but this time to Spiros. Because you explained to us there's quite a lot of heterogeneity in diseases and phenotypes and how they're expressed. What potential or risks do you see in digital twins of humans in mapping diseases? So the question is, what types of humans or what human is mapped? How generalized is this? And how would this come together or not come together with approaches to personalized medicine?
Yeah, so I'll be honest and say that the digital twins is something that I don't fully understand, unfortunately. So even the fact that there's a digital twin of the heart kind of blows my mind in terms of how it was developed. I don't think there's any such thing as the average human or the average patient for a particular disease. And I think part of that is reflected in the heterogeneity. So I don't even know where to start to kind of address that in terms of, other than characterizing current disease states as much as possible. I mean, you probably know much more than me but
yeah, so this digital twin concept is actually quite a promising concept, but it's really a long time, or many tasks ahead of us in order to actually make the in silico models as in the way as they should be in order to make the digital twin. But the concept here is that if we will be successful in the future, that we do have for organs or maybe for specific subcell populations, digital representations for a particular patient, for a particular time point, learning, then maybe and mapping this information to a database of digital twins of formerly of patients which we have seen in the past, in order to decide and assign what would be the best treatment option for this newly seen particular patient. This is the hope in these type of models, which is a big hope, of course, but from the computer science perspective and also in silico modeling, we have many, many things to do in order to fulfill this hope.
Thank you. Spiros, actually, to be honest, I was wondering why somebody like you who had such rich data in London would come to Austria, as the Austrian data landscape is quite of a nightmare still. But I mean, I get it. And there's a lot of building up right now. So I was thinking, which data could you use? Because actually, we've been working with social security data in the past, more from an economic perspective, but it was kind of messy and difficult to treat. So there's these other projects going on where we are currently linking survey data with registry data from Austria, from different registries, and these are currently building up, but there are interesting registries coming, and they're not only from the economic ministries, but also from the health ministries. And then there is other ERICs in Austria, like the Genome Project, which is currently looking for funding, and they are sitting in Innsbruck. And then there's the BBRI, the Biobank Project, and so. So all of us, we are trying to put it to make our data linkable to registry data and interlinkable between the different projects. So I would think maybe this would be a future database for you to carry on your research, which would also have the advantage of not being so biased as if you went directly to a hospital and only retrieved hospital data from this specific hospital, since you only have a limited subpopulation in that hospital compared to more general. I don't know. What do you think?
So these are sort of So first of all, most of the data that I use actually is data that's open access. So people can apply from anywhere in the world and get access to, with the exception of the really large one. I think that the fact that Austria has a fragmented data system is both a challenge and an opportunity, because it can only get better. And but I sympathize, because it's sort of the same in Greece as well. I think the way I would approach it is try to show value in using the data to answer very, very simple questions to begin with, rather than try to do something very complicated cross-sector and then build on top of that. So there's definitely a lot of stuff that you can do, even with sort of data that's across two sectors or across two sources and then linked with public data. So I'm optimistic that it can work. And there is definitely, when I was doing homework for the interview, there's definitely appetite to digitize things, link things, reuse things, and integrate them in the healthcare system. So it's definitely something that's going to improve. So I'm optimistic. But if you do have any specific sort of projects and data sets, yeah, please get in touch. I'd love to learn more, because I'm still kind of learning about everything.
May I add something to that as well? So because of this particular new type of treatment, which is not available to many patients, this CAR T-cell treatment, it's also not available to a huge number of patients in the same hospital. And because of that, I'm also one of the founding members of the Itu.austria Association, where one of the tasks is to establish federated networks, federated learning infrastructure to support it in a sense, to make data in a federated style available also for training of machine learning models and so on.
Since the pharma industry is such a profitable sector, I'm wondering, what is your relation, the relation of your research, your basic research, with the industry? Are you far away from application, or is industry basically doing the same and offering you a job every other week? And a follow-up question, maybe, what is your eye on global pharma and medical application and industry? Is that one of the sectors where Europe can be competitive globally? But this is just a follow-up, and I know it's going somewhere else.
So no, I will stay at ITU, so don't worry about that. But so pharma is a partner for us. So there, from the European scale, we do have many projects together with big pharma companies as well, formerly called IME2, but now IHI Quant. And given this setting with these FPR partners, we co-develop things, and they are quite interested what we do. Yeah.
Yeah, same. Lots of projects working with partners from the pharmaceutical industry. I mean, we have a really long-standing collaboration in my lab with GSK, and we're sort of their kind of phenotyping partner. So and again, it's kind of that comic that we develop algorithms to define diseases, which they can then apply in their internal trial data sets or in their internal analyses, which, and then they show us how they're used, and is, quite gratifying. So I think it's a collaboration in that sense, and we co-author papers, and it's good. And to be very frank, they can do things at a scale and a speed that university partners would struggle. So I think I'm happy with how it works. Obviously, there are caveats to any collaboration in terms they have different priorities, speeds that they work with, all these things. But I think it is what it is.
Yeah, super quick follow-up. So you say your collaborators, at the same time, of course, they can lift much higher investments and scale things. So is there a kind of value chain hierarchy where you are more on the basic research side and you do some developments, and once something promising shines up, they they pick it up or even buy it from you? So it's basically two questions. Is there a hierarchy in the value chain? And secondly, do you create value that is actually bought by industry?
Yeah, so one of these biomarker sets I've developed in the last years is now with a company that they develop it to until the very last end. But for the general, so for the CAR-T field, this is still quite emerging and new field. So here we collaborate and talk to pharma partners because they learn what we do because we do it with the real world data, and this is not accessible to them in an easy way. Yeah.
Yeah, same for us. I mean, I think on the drug development pipeline, we're further ahead. So we're not with the basic science. We're more in the sort of later stages of translation. So they look at the things we work with, and they can inform trial designs or efficacy studies and things like that. And the way we work, again, as part of that collaboration, is they have a embargo period where we give them, we collaborate and they can use their results for 1 year, and then after 1 year we open source everything and we publish and we make everything available. How they're, so it's harder for me to say, oh, this particular molecule was discovered and then it got sold, and that's how they used it. So it's not to that scale, not to that mechanism, at least for the stuff that we do.
Since we are the interdisciplinary university, and you obviously are very interdisciplinary people, I just wanted to know, when did this interdisciplinary virus hit you? And how did you deal with it?
So to be honest, it actually already hit me in the very beginning of the first day of my studies, because I chose to study computer science, but I always from the very first day knew that I will apply it to biology.
Yeah, same. I think we have really similar profiles, actually. I mean, my PhD was on yeast, so then I thought, oh, yeast is exciting, but maybe humans are more exciting, so then I joined UCL, and then you're working with clinicians and...
So that was already predetermined.
Yeah.
The moment where you studied computer science.
Yeah. Okay. Yeah.
Cool.
Can I say I don't see any further questions on the floor? I would ask, are there any questions online that we are aware of?
Yes, there is one. Do you want to read it?
There is one online question. Why would it not be possible for the family or person be treated to be able to give the access to the medical data? Why would it not be possible?
I think this is about data donation, maybe?
Yeah, or from what I understood from the question, are there any challenges, like getting the patient data, or kind of making it accessible to the patient itself?
So if we work with molecular data, it's always a challenge actually to get access to the data, because we need, of course, because this is per definition sensitive data. And here we need to follow strict legal rules like GDPR and whatsoever. And what my experience is actually that, to be frank, sometimes not all the process has been set up in a hospital, such that when I collaborate with or start a new collaboration, sometimes we need to reset up everything, all the paperwork in the hospital to make us, to make the data also accessible to us as an external partner, which is a huge amount of work. But given the mission and the vision behind it, what it actually can mean for patients if we make the data accessible, then I'm happy to actually conduct this type of work as well.
Yeah, same for us. I mean, there are two routes. It's either consented data, where someone has consented to take part in a study, and then data is collected, or unconsented data, where you can go down a legal route and say this is for the public benefit, so we can use this data, obviously without the sensitive identifiers for research. So consented data is usually smaller in size, in sample size, but deeper. So you get much more detail, because you invite individuals and you do tests and you genotype them, you sequence, etc. Unconsidered data is much wider, larger in scale, but shallow because you get data as part of payments or as part of like routing things rather than research. So it's both are equally good, but all equally challenging to get. Yeah.
Thanks for the response. I hope that we hit the nerve of the online question. Also, it's an invitation to join next time at the presential event. And with this, I'd actually like to close the stage event, so to say, and start our networking event afterwards. Thanks a lot to all of you having been here. Thanks a lot to the online audience, and of course, thanks a lot to our two speakers. I would like to invite a last warm round of applause, and then I'd like to see you afterwards.